In this virtual tour, you will visit two sample prep labs, the School of Earth Sciences Clean Lab facility, and our thermal ionization mass spectrometry, or TIMS lab. Many of our students and research scientists use these facilities to process and analyze samples, ranging from ancient microfossils to deep sea clays to surface waters from our backyard all the way from Antarctica. If you are worried about contamination of a sample, I'm sure you'll find your way to our clean lab facility. Rock samples, primarily limestones, are the focus of much of Dr. Saltzman's group, as well as microfossils, which can all be processed in-house in his lab in Orton Hall, just below our museum and dinosaur. Soft sediment samples, primarily from ocean drilling, are the focus of much of my work, which can be processed in what I refer to as the dirty wet chemistry lab in Mendenhall. Sediments are requested from the International Ocean Discovery Program over the particular time interval of interest and are processed here. Brandon is a master's student extracting the mineral barite from Pacific sediment, which involves over a week of work in the lab exposing the sediment to various reagents to digest all the sediment except the mineral of interest that is concentrated in the residue collected on the filter you see here. Now we will visit our clean lab facility just down the hallway. Here we process samples in a particle dust-free environment under positive pressure filtered air. Some samples dissolve easily in acid, and others, including refractory minerals, clays, and dust from ice cores, require a cocktail of dangerous acids. Once a sample is dissolved, we can separate the elements of interest and even concentrate and purify specific elements so that they can be analyzed by our mass spectrometer to determine the ratio of isotopes in our samples. To do the separations, we do column chromatography, using specific resins depending on the element of interest. Currently, we have users separating calcium, strontium, neodymium, and rare earth elements. A lot of the time spent here is for cleaning, distilling acid, cleaning vials, pipette tips, and even vacuuming the ceiling. Once we have purified our sample, we can pass it through our TIMS lab. We enter the lab through some of our student offices, which are currently very quiet due to COVID restrictions. Here we load each purified sample and isotope standard onto metal ribbon filaments. We fit them to a sample wheel and load them into our sample chamber. During analysis, each sample is heated to over 1,000 degrees Celsius. When the sample evaporates and is ionized, it can be focused and accelerated through a flight tube where it is passed through an electromagnet that separates the masses of the ions based on their mass to charge ratio, and eventually measured in one of our 9 movable Faraday cups. We can also count individual ions in our center cup for very small signals. Thanks for visiting our labs. You can check out our websites, send me an email, or stop by the lab when you're on campus for a more detailed tour.